Mm. Excellent. So, can you guys tell me the 4 types of bonds? Now, don't all of you guys talk at once. There we go. Let's see, we have polar, we have nonpolar, we have, both of those are covalent, right? Polar, nonpolar, there are 2 forms of covalent bonds. Ionic and I kind of criticize that very last one. I keep saying it's not like really, there we go, hydrogen bond. So we have polar, nonpolar, both of those types are covalent bonds, and then we have ionic bonds, and then we have hydrogen bonds. Which one is the strongest bond? Okay, how about which one is the weakest bond? Hydrogen, because hydrogen is more like an interaction, where covalent, they're overlapping. So you mean they're sharing electrons. That's going to be pretty strong, right? And then also ionic is a pretty strong reaction, as long as you don't stick to one. Right? Okay, questions, concerns, Gash? Okay, so let's see what else. What else did we talk about? So, didn't mean to put this up. Can you guys, what are you guys seeing? What do you guys see? That wasn't supposed to be up yet.
Yeah.
Okay, so if I'm using monomers to build a polymer, particularly in the body, what type of reaction is that? First off, just generally the name. And then we'll go specific. Anyone? Anyone? Can anybody tell me the different types of reactions? Anybody tell me the different types of reactions? Adrian, synthesis reaction. Yes, synthesis reaction is one of them. Yes, good job. So that's one. The exchange reaction, that's 2. Any others? Think about life. How many of you guys, did you guys eat today? Any of you guys eat today?
Yes.
What did you eat, Jesse?
Rice with eggs and spinach, and Alejandro's eating right now.
Alejandro is eating right now. What are you eating, Alejandro? Chicken, right? Okay, chicken is a good example. So Alejandro is tearing up some chicken, right? What's happening to that chicken as Alejandro tears it up? Oh, he's destroying it, right? Okay, so what is another term that we can use for destroy? Break down. Okay, he's breaking it down. So there's another term. There's a term that we use for particular reactions. If we break things down, we starts with a D. So if we want to get rid of bodies, what do we do? Obviously, we don't have true criminals here, right? That's a good thing, right? Because we don't know how to get rid of bodies, Jesse. So what do we, should we teach them how to get rid of bodies?
Sure.
right? So we want to decompose our body, right? So what type of reaction would that be then if we're decomposing? Anyone? Adrian. Boy, this class got small.
I know. From
The
8.
morning class got big. This one got small.
Yeah, I wanted to get it out of the way.
Yeah. So a decomposition reaction. So we have synthesis. Synthesis means building. And then we have decomposition, right? Breaking down. Okay, so if we talk about our metabolism specifically, right? We use water for a lot of things. So if we get rid of water in a reaction, are we going to be building? And what is that process called of us getting rid of water? And is it building? Okay, I already got my questions now. This is making it easy for me. That's question.
Oh, God. Does it? Oh.
Okay, yeah. Antibiotic means building. Can't talk today. Catabolic means breaking down, right? But the process of building by the use of water, what process is that called? Any guesses? Soledad, any guesses? This is a really talkative group. Yeah, it does evolve kind of hydrogen in a sense because water is in hydrogen. So we'll touch base. We'll start there then. You guys remember this slide? So dehydration synthesis, where we basically remove water to combine two monomers to make a polymer. Monomer one here, monomer one here. We take an OH and an H from monomer one and monomer two. We get water. And then as a result, we get a linkage between the two monomers. You guys remember that?
Yes, Dr. Henry.
Thank you. Good, good. Okay, so then we break down a polymer by adding water. And we call that hydrolysis. Hydro meaning water, lysis meaning breaking down. Make sense? Okay, so I want to fit into my new prom dress, right? I picked on a little, I got what, COVID 35. Okay, and I got this really, really cute prom dress. And I want to try to get back into it. So am I going to drink a lot of coffee? Or am I going to drink a lot of water? And why? Anyone? Boy, we're going to have a really good test. I feel it already. Do you feel it, Jesse? It's going to be a good one.
I feel it deep down.
So, anybody want to take a guess? Okay, Alejandro, give me a guess. I want to drink more water, right? Because I want to break down all that fat. Is that true? Okay, so by drinking more water, it allows us to sit there and break down things more frequently, right? And it'll break down some muscle, but it'll also break down our fat as well too. And then it also does another job of flushing some of that fat out, right? So it's a twofer. Okay, so let's talk about the dissociation of sodium chloride. Man, my phone is on fire. So sodium chloride is a crystal. What happens when you put sodium chloride in water? When you put salt in water? It starts to dissolve. That's right. So let's talk about why it dissolves, right? We know that salt, sodium chloride, is an ionic compound. It has a cation and an anion, right? We know that water, so that means that it's being held together by positive and negative charges. Sodium is positive, chlorine is negative, comes together. Kind of like we did when we were all mushy-mushy with our boyfriend or girlfriend who were total opposites. Right? Ooh, yeah, I love you, right? So in the case of water, water is, it has two poles. Oops, I carried away. It has two poles. It has a partial positive and a partial negative pole. Hydrogens, the hydrogens on water are partially positive. The oxygens on water are partially negative. They're weak positive charges in the case of hydrogen, and they're weak negative charges in the case of oxygen. Where sodium is a strong positive charge, right? So water does this. Water goes up to sodium, you know, as chloride just kind of has its turn back, his head turned a little bit, and says, hey, baby, you don't need chloride. You got me. I'll treat you good, right? So then water basically surrounds the sodium, that plus charge. Now, it's the oxygen part of water that surrounds that sodium because it's partially negative. So those partial negatives interact with that positive charge. So sodium is like, it's all blushy and everything, right? Because water's giving her attention, right? So then soon as sodium, you know, is kind of occupied and a little distracted and everything like that, water goes up to chlorine and says, hey, dude, you don't need sodium. I got you. I can take care of you, right? So in this case, water interacts with chloride, the negative charge, the negative ion, right? By its partial positive charge of hydrogen. And it surrounds the chloride ion, right? So then it separates the two, right? Water's a home wrecker. You guys get that? You see it? Pretty simple, right? And so because water surrounds both of them, it keeps it in solution. It allows them to be separated, dissociated, right? And so water uses the fact that it's polar, which allows it to make two poles, right? That positive pole and that negative pole to sit there and keep things in solution. It does the same thing with alcohol, right? How many of you guys turn up on the weekends? Hey, gotta get my drink on, right? So when we turn up on the weekends, often we'll sit there and have, you know, our nice little drink with a couple of things of ice. And if we let that ice sit for a long time, it gets all watery and stuff like that. And if we stir it, we can't tell the difference between the water and the ice, right? The water and the alcohol. Because the water will actually start, or I should say, the water will suspend the alcohol in solution. So then that way we can't see the difference between the alcohol and the water. It looks the same to us, okay? Because of the same reasons. So in the case of alcohol or ethanol, it has carbon-carbon, hydrogen, 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 hydrogen. Oops. Okay, oxygen, hydrogen. So water comes along like this. Being partial negative on the oxygen and interacting with, in this case, partial positive of the hydrogen. And then water comes along and does the same thing here. But the hydrogen is going to interact. And so it keeps it in solution for the same reasons, because of those partial charges. Okay? Questions, concerns, Cash? Good, no questions, we move on. Okay, so have you guys heard of acid and bases? Yes. Okay, so we're going to talk a little bit about acid and bases as well too. So when we talk about acid and bases, they're also ions, right? So an acid is an ion compound that has a hydrogen ion, and let's say if we're talking about hydrochloric acid, we have a hydrogen ion here. So this becomes, when it becomes in water, AQ means in water, it becomes H plus, because water separates those ions, plus Cl minus. Okay, so in that case, water is going to be surrounding that hydrogen, that plus in that case, and water is also going to be surrounding the chlorine, kind of like you did with sodium, and allow it to be in solution. Now, acids are defined by how much H plus they have in solution, right? So when you guys heard of pH before, right? pH 7. Well, pH is just minus the log of the concentration of H plus, or the hydrogen ion, okay? You guys know what I mean by concentration? Anybody? Concentration. You guys know what I mean by concentration? Yeah. Good. Okay. So how many of you guys, the amount there is? Yeah. So it's the amount of stuff that we have in a solution, right? So a high concentration of H plus means that we have a lot of H plus in solution. A low concentration means that it's dilute. So that means that we have a little bit of solution. How many of you guys used to make orange juice from the little frozen cube of orange juice that you get from the store? Isn't that stuff called concentrated orange juice? Concentrate, right? And then you just drop that into a big old tub of water. Then you get some orange juice. Anybody? Or am I dating myself? They don't do that anymore.
And I know what it is. I just never used it.
Oh, okay. And then exactly, and then dump a ton of sugar in it because it's always really bitter, and so you got to sweeten it up too. I'm right there with you. Okay, so this is how we would calculate our pH, right? And so we have this nice, beautiful scale called the pH scale. It's based upon how many H plus molecules that we have in solution. The more molecules that you have in solution, the lower your pH is going to be, right? The more of the H plus you have, the lower the pH is going to be. The higher the pH, the less you have in solution. So we also have what we call a base. So like sodium hydroxide is an example of a base. These guys are also going to form ions. So in this case, we'll get Na plus OH minus. Again, in water, EQ. AQ just means in water is. It stands for aqueous. And so we can also calculate what we call the pOH, which would be the minus the log of the concentration of OH, right? And this kind of gives us an idea how the base is. But we have this nice, beautiful scale because we know that water, H2O, which can sometimes act like it's an acid and form that H plus and that OH minus. But it's going to be equal. Water is neutral. So if we calculate the pH, it's going to be a pH of 7. So it's like that halfway in between. So it has the same amount of hydrogen ion as it has hydroxyl ion. Okay? Are you good? Okay. So pH of 7 means it's neutral, right? So we're balanced. Water is always neutral. It's a good place to be at. So if it's lower than 7, it's acidic. That means there are more H pluses. Okay. If it's above a 7, then there's more OHs. Okay. So I'm going to show you a pH scale. Clear. So this is a pH scale right here. Here's our neutral, pH 7. So pH 6, pH 5, pH 4, pH 3, pH 2, pH 1. So do you guys know something that is acidic, that has acid in it? Oranges, yes, oranges. Lemons, perfect, yes. So orange juice, lemon juice, both of those are acidic, right? So they have a lot more H plus than they have OH minus. So lemon juice is about a pH of 2. Orange juice is about 3.5, somewhere in there. Okay. I drink coffee every morning. My coffee is acidic. It's probably the reason why I always end up having heartburn, right? Because I have my stomach acids all pushing up. Have you guys heard of acid reflux? Yeah. So all it is that the acids in your stomachs are starting to come up your esophagus. So what is one way to kind of neutralize an acid? Water. Well, water doesn't really neutralize it. It just makes it a little bit of a weaker acid. Alka-Seltzer does, because Alka-Seltzer actually has something that is a base. So Alka-Seltzer would be basic. So the way you would neutralize an acid is by adding a little bit of base to try to neutralize it. So in that case, often I will sit there and take a spoon of baking soda, right? To kind of neutralize my upset stomach. Okay, so then that helps me out and my stomach settles down a little bit because baking soda is basic, where my coffee is acidic. So it kind of helps neutralize my coffee. Maybe I could just, save that, doing it in my stomach and just put a, spoon of baking soda in my coffee in the morning. Should try that. Okay, so some other things that are around the house that are kind of basic is bleach, ammonia, liquid drain cleaner, right? So all of those things are basic. Seawater is basic, right? Where regular water is neutral, right? Your blood is a little bit basic, or should be. Your pee is a little acidic. Your milk you guys drink, it has lactic acid, it's a little acidic. Does that make sense? Okay. Okay, so now we're going to talk about the biomolecules of the body, the major biomolecules of the body. The first one that we're going to talk about is called carbohydrates. Let me find my pen. Carbs, or we like to call them carbs. Okay, carbohydrates. So there are a couple of types of carbohydrates. One of them is called a monosaccharide. Mono means one, right? So that means it's only one sugar. That's what monosaccharide means. So these are examples of one sugars. So glucose, fructose, and galactose are all examples of hexose sugars, meaning that they have 6 carbons. Hex means 6, right? And it represents the carbon. So each of these corners represents a carbon, except for this one. This one actually has some oxygen in that corner. So that's 1, 2, 3, 4, 5. And then we have that carbon that's poking up here, 6. This guy also has 6 carbons. 1, 2, 3, 4, 5, and this one poking down, 6. This guy also has 6 carbons. 1, 2, 3, 4, 5, 6. So let me give you the chemical formula for all of these carbons. We have C6H12O6. That's the chemical formula for all of these. Do they look exactly the same? Adrian? Yes, no, maybe? No? Yeah? No, they don't look exactly the same. This one has 1, 2, 3, 4, 5, a 5-ring carbon. I mean, a 5-ring structure. This guy has a 6-ring structure. This guy has a 6-ring structure. This guy has the O's going down. This guy has the O's going up. Okay, so glucose and galactose are really close, but they're just facing different ways. They look similar, but they're different, right? So these guys are called isomers of each other. They have the same chemical formula, but they have different chemical structures. Different chemical structures, but same chemical formula. Isomers. Okay, so another example is here we have, well, it's not quite the same. This guy is called deoxyribose. This guy is called ribose. The difference between deoxyribose is that deoxyribose gets rid of 1 oxygen. That's why it's called deoxy. Means I removed 1 oxygen, right? So we know that deoxyribose plays a huge part in nucleic acids, and we'll be talking about that a little later. So these are all 5 very important monosaccharides that are going to be found in the body that you should be familiar with. Okay, so these guys are called disaccharides. Why do you think they're called disaccharides? Di. For 2, exactly. So disaccharides means that they're 2 sugars that are brought together by dehydration synthesis. So we use dehydration synthesis to make these disaccharides. One of the most famous disaccharides is table sugar. Table sugar is called sucrose. It's made by combining glucose and fructose and removing water, dehydration, right? And they come together by that oxygen, okay? Another one is lactose. Where does lactose come from? Milk. Good job. Yeah, so lactose is made up of galactose and glucose. And together they form lactose. Again, it's made by dehydration. The last one is maltose. How many of you guys have malt shakes? Malted shakes? Delicious. Okay. That has maltose in it. Those are just 2 glucose molecules connected together. You've never had one? Oh man, you're missing out. You need to go to Superior Dairy in Hanford. Bomb malted shakes. You know, Jesse's writing it down. Superior Dairy in Hanford, malted shakes, you know.
I already had one. I had one from there. It is good.
Okay, it's expensive, but it's really good.
It is, yeah, but I liked it. It was enjoyable.
Okay. So our body, we actually eat a lot of sucrose. How many of you guys eat candy? All the time. We drink a lot of milk, right? So our body actually breaks these guys down using enzymes to make it happen faster. But when it does the breaking down, it goes through hydrolysis. We have to use hydrolysis to sit there, break them down so that we can use the glucose. Glucose, we use glucose first, and all the other sugars have to be modified or changed so that it becomes glucose before we can use it. Okay, good. Man, this is an easy class. 3 people, we should have more classes like this. Now, if I only had the grade 3 test, that would make it so much better.
Yes.
Okay. So far we've talked about monosaccharides, we talked about disaccharides. So the next one is we call polysaccharides. Polysaccharides are long, really long chains of sugars that are brought together. And again, these guys are connected by covalent bonding, right? So we have amylose. Amylose is just a very long chain of glucose that is connected, right? So it's just glucose, 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 glucose. But it's a single chain. It's not branching or anything like that. That's amylose. So amylopectin is a branching chain of sugars. Both of these guys are considered starches. So do you guys eat flour? You make bread? Right? You eat potatoes? Love me some potatoes. Do you eat corn? Oh yeah, love corn. What is the corn? Alethos? Is it ELOT? How do you say that? The corn with
Elote.
elote. Thank you, elote. Yes. So elote. Oh, I love elote, right? So those are examples of starches, right? You get a lot more amylose in corn, right, than you're going to get in potatoes. Potatoes are going to be more amylopectin. Elote is going to be more amylose, right? And so that's how you get a little more of that sweetness, because some of those sugars get broken down and you just have that sucrose that makes it real sweet. Have you guys ever just ate flour alone? Just popped in your mouth. No? Am I the only weird person that does things like that? I used to, as a kid, I put everything in my mouth. Popsicle stick, you name it, it would all go in there. You never chewed on a popsicle stick and just... Okay, so in case of flour isn't that sweet, right? So that has a lot more of the amylopectin because it has those branching chains. So it takes a little bit more to sit there and cut it up. Oh, popsicle stick, but not dried flour. Okay, we'll get to that popsicle. Okay, so the other one is glycogen. Glycogen is found in our muscles, right? Our muscles actually store glycogen for food for later, right? So again, these are just long chains of glucose and all of these branching structures. And so they put that in our muscles, and so we get glycogen stored up in our muscles, okay? So we do this, plants do this, they make starch, okay? The other thing plants do is cellulose. So that popsicle stick is cellulose. So the way fiber works, how many of you guys eat something that's fibrous? Fiber. Right? You guys eat veggies? Yeah. So a lot of our fiber comes from fresh vegetables and stuff like that. Or you can eat bark, one or the other. So bark will work. So in all of those cases, our body just basically is not able to break down the fiber. It's not able to break down that cellulose, right? Because the way these chains are made, we don't have enzymes that are capable of cutting it the right way. So we can never get any of that sweetness, but we chew the crap out of popsicle sticks. And if you actually swallow it and eat it, it comes out in your poop. Does it not? Because you can't process it. You guys all eat corn. You guys know that if you paid attention and you ate a whole bunch of corn, and then you looked at your poop the next day, you also see the husk of the corn, right? The little yellow part of the corn. That comes out in our poop because that's cellulose, right? So those are forms of fiber. And the way fiber works is, as we eat it, our body isn't able to digest it. So it just keeps pushing it out. It's like, you got to go, because we can't use it as a food source. So it just goes through us. Make sense? Good. Okay. So those were the carbohydrates. So now we get to talk about the lipids, fats, right? So, oops, I got carried away. Oh yeah. Okay, so first we have what we call triglycerides. The way that triglycerides are made by dehydration synthesis. Just the same way that The starches were made the same way that the disaccharides were made, again, by dehydration synthesis. So we basically bring two things together. In this case, we're going to bring glycerol and the fatty acid chain together. And so, again, we're going to remove water. That's what happens here. And as a result, we get triglycerides, or we call them neutral fats. You know, that fat in my belly, you know, that's that fat, okay? The fat I need to get rid of to fit in my prom dress. Okay, so fats, the way that they kind of are, let me give you a little more information, is that they have these long chains of carbons. These carbons are non-polar, right? So since they're non-polar, they're not going to want to interact with water. They want to stay away from water. They're hydrophobic. Have you heard that term before? Hydrophobic? Yes, you're right. My fat does provide an extra work. And as I tell people I'm dating, you know, it says it's more cushion for the pushing, you know? So in hydrophobic meaning they're dissolving in water? No. Hydrophobic, what does phobic mean? If I have a phobia, fear, right? So hydro meaning water, so hydrophobic means fear of water. Make sense? So these long chains are scared of water. So if we put fat, have you guys, what is the fat called? Right now I can't remember anything. Starts with an M. There's a big old tub of it that you buy. Starts with an M. Like you use it to make tortillas. Is it called? Maize is corn, isn't it? But it's something like
Maize.
Maize.
Yeah.
So
it's corn flour.
Not corn flour. I'm talking about lard. Fat lard.
Oh,
It starts with
manteca.
Manteca. Yes, thank you. Manteca. So, okay, so I have to tell you the story. And it's a total aside. So I used to live next door to these women, and they would have these little get together. We'd watch, I can't remember, I think it was Grey's Anatomy or something like that. And we'd all come together, and then they would cook. And one of them, she would always make her tortillas with manteca, and then the other one would always make her tortillas with like canola oil, right? And I hated coming over when the one was making with canola oil because it tasted horrible. I don't know what it was about that vegetable oil. It just didn't have that same taste as that manteca, right? And so there is a distinction between those two, right? One is in a liquid form and one of them is a lard. And we'll be talking about that lard and how they differ from each other in a second, okay? So in this context, right? So we know that if we put a big old chunk of lard into water, it floats on top of the water. It doesn't dissolve. Is that true? Why? Why doesn't it dissolve? Because it's hydrophobic. Exactly. It's hydrophobic because those non-polar components of carbon, they're non-polar. They're not, they're neutral. They don't want to sit there and have any charges associated with them. They're not going to interact with charges. They're kind of elitist. No charge for me. You stay away from me, right? So non-polar. So that's why oil and water don't mix, right? Or oil and vinegar, right? Okay. So one of the lipids are triglycerides. So triglycerides are basically our fats, okay? So we have two types of fats. One of them is called saturated fat. The other one is called unsaturated fat. So saturated fat are the ones that is the lard, right? So unsaturated fat is like our canola oil. So saturated fat, It's basically all single chains. There's no double bonds or anything like that. So they're all single bonded between each of the carbons, meaning that they only share 2 electrons. This carbon and this carbon only sharing these 2 electrons. This carbon and this carbon only sharing these 2 electrons. Where a unsaturated fat will have at least one. It can have more than one, but at least one double bond or one triple bond. So in this case, these guys are sharing 4 electrons. Now, what happens with unsaturated fats is they form kinks. They're not straight, so you'll get little kinky structures, right? Because you're getting those kinky structures, you're going to be a liquid more so than you're going to be a solid, because these guys are just going to be able to lay like right on top of each other and interact directly with each other. And carbons are kind of elitist. They're like, oh, I don't interact with oxygen unless I absolutely must interact with oxygen on another atom. So if you have a long chain of carbon, it's going to want to interact only with other carbon structures or non-polar structures. So all of these guys, so you'd have chain on top of chain, on top of chain, on top of chain, on top of chain, on top of chain. And so that large is all solid. They all like each other. So all of those hydrophobic components get together and sit right on top of each other. And so in the case of oil, what happens is that you have kinks. And so you can't get that complete laying on top of each other. You kind of get those open structures. And so it gives it a little more fluidity. And so it makes it more of a liquid, more so than a solid. Does that make sense? One day I'm going to draw a real beautiful picture, put it on my slides. Okay, so which one is supposed to be unhealthy for you? Saturated fats or unsaturated fats? Saturated fats, right? Because they cause cloggage, artery cloggage, because they just kind of lay flat on top of each other. So they just kind of build and build and build and build, and then they clogs up that area. Okay. Okay, so we've talked about saturated and unsaturated. So let's talk about some more lipids. So we have what we call the phospholipids. The phospholipids are lipid structures that have your fatty acid chains. It has the glycerol backbone, and it also has what we call a phosphate containing group or a phosphorus containing group. Okay, so, and that's how we get the term phospholipid. Okay, because it has that phosphate. This is phosphate. Okay, so phosphate, because it has all these oxygens, and also because that phosphorus, it makes it more polar. So this guy has what we call a polar head and a non-polar tail. Okay, so this guy loves water, and this guy hates water. So phospholipids are found in your cell membrane, and so they form what we call the lipid bilayer. And so when they do that, this is how we just kind of draw it simply. You guys can see it right here. So that would be my hydrophilic head, meaning it likes water, or my polar head. And this is my hydrophobic tail, meaning it doesn't like water. So your membrane does this to form, and you guys will see this in the next chapter. The hydrophobic tails come together. And then here, this will be our water out here. This is water. I shouldn't, let me see if I can do it better here. My hydrophobic tails and my water are looking the same there. So this is supposed to be water out here. This is water. Okay. So our our polar heads are going to want to touch this. Hydrophilic heads. Our hydrophobic tails, they're going to point inside. And because they do this, they're able to sit there and form a membrane that doesn't let water through. Right. So water can't get through our membrane because we have these structures. Instead, it has to use what we call a pore. And so it makes these protein pores called aquaporins to let water go in and out. But that's regulated. So we don't want too much water to go inside the cell, because otherwise the cell will blow up. We don't want that to happen. So we want to kind of keep it controlled. We want some of it on the outside, and we want some of it on the inside. The cell, this is our cell, it has water on the inside as well as it has water on the outside. Does that make sense? Or am I just talking crazy? And so your membrane, because of the structure, that hydrophobic part of it, it keeps that water out. So water can't pass through here. Okay? Okay, so steroids. You guys know what steroids are? Not the stuff you shoot up in labs, to get buff or anything like that. What we're talking about is like cholesterol. Cholesterol is a steroid. And so cholesterols are often found in membrane structures too. They also lead to the fluidity of the membrane. So your membrane is constantly turning over, meaning that it changes, like it gets a little old, and so it'll take those parts and then recycle them and rebuild them and then send it back up there. And so it's constantly moving. So there's parts of your membrane that's rigid, and there's parts of it that's constantly flowing. And cholesterol is responsible for that rigidity as well as the flowing of it. And we're going to talk about this a little bit more. So we have steroids, and this is an example of a steroid going on right here. Oh, Lord. Okay. And then our last lipid structure is called prostaglandins. Prostaglandins. So prostaglandins, these guys are, they're going to be derived from unsaturated fats, essentially. Okay. So they basically will have double bonds or triple bonds. And again, we'll talk about them. A lot of them are involved in signaling processes. Okay? Are we good? Good. Okay. So we have lipids, and we talked about carbohydrates. Next, we're going to talk about polypeptides. So polypeptides are made up of the building blocks are amino acids. And an amino acid is composed up of, it has an amine group, it has what we call the side chain, and we have a carboxyl group. So carboxyl group is this carbon double bonded to an oxygen, and then it's also attached to an OH. So that's called carboxyl. So this is the carboxylic acid component. This guy is called an amine. It's called an amine because it has the nitrogen. So anytime that you see the term amine, that basically means it has a nitrogen. Okay? And this side chain, this R basically means it can be changed for different things. So your body has a total of, do you guys know how many amino acids are in your body that you guys use to make protein? Anyone? Anyone want to take a guess? 5, 10, 15, 20? 20. Yes, there are 20 amino acids that are used to make up your protein structures. So the difference between each of those amino acids is that side chain. The side chain acts as a difference. Each amino acid has a different side chain associated, and it gives it its characteristics. So if it's a long carbon chain, it's going to be hydrophobic, right? So if it has an SH on it, it's going to be hydrophilic. If it has something with an OH on it, it's going to be hydrophilic, meaning that it likes water. Okay, hydrophobic, hydrophilic. So some of them are going to be like plus charges, some are going to be minus charge. So just depending on what it is, it's going to have different functions, right? And that plays a part in organizing the protein itself. We'll talk about that here. Okay. So when two amino acids come together, they do so by peptide bonding. This is done by Guess what? Anybody want to take a guess? Starts with a D. Not everybody yell at once. Okay, so I guess I'm just going to yell it. Dehydration synthesis, right? So in this context, this is one amino acid, this is another amino acid. We remove that water and we form the peptide bond. Okay, so that's how we build up proteins, is by dehydration. Okay, so if I want to go out and be buff, right? Because proteins, muscle, isn't that made up of protein? So now I no longer want to, be in my little tight little dress. I want to be ripped, right? So now what I'm going to do is I'm going to kind of dehydrate myself a little, right? Because that'll help that synthesis of muscle, building up that protein by having that dehydration, right? So it helps induce this process. Make sense? Now you don't want to dehydrate yourself completely, because part of muscle building is the breaking down as much as the building up. So it's a combination of both. You got to get the right equilibrium. Okay. So a peptide bond is just a bond between two amino acids, that is forming between two amino acids, or between two peptides, long chains of amino acids, or polypeptides, or protein. Okay. So let's talk about those proteins. So we basically will form peptide bonds to form proteins, right? So when we talk about our protein, we have what we call the primary structure. The primary structure is just the sequence of amino acids as they make up the chain. So this will be one amino acid. This will be another amino acid. So this could be like glycine. This will be tryptophan. This will be histidine. This will be, so there'll be different amino acids, okay? So now these amino acids, this primary structure is going to be used because they're going to be interacting with each other. So each amino acid could interact with another amino acid on that chain. These guys are covalently bonded together. These guys are going to be interacting by hydrogen bonds and by van der Waals interaction, or the fact that things are hydrophobic. Two things are hydrophobic, they're going to want to interact compared to something that is hydrophilic, right? Or polar, right? So they're going to be using that polarity to determine whether or not they're going to interact with each other. And they're going to form what we call either an alpha helix, meaning that you guys, like girls with really, really curly hair, those little curls. Yeah, no, maybe. Jesse, you know about girls with curly hair. Yes, because your girlfriend has curly hair, right?
Yes, I do.
Right? Okay, so that's what an alpha helix kind of looks like. It's like that little loop, right? It's like a helix, a curl, okay? And then the other thing that they form is called beta sheet. This is an example of beta sheets. Beta sheet is like a folded sheet of paper. So you're going to either form an alpha helix or a beta sheet, and that's done by your hydrogen bonding, and then also just hydrophobic interaction. So that's going to form what we call our secondary structure. Okay, so secondary structure is your alpha helix or beta sheets, or a combination of both. Okay, because you could have an alpha helix and then later on have a beta sheet. So then your tertiary reaction is just kind of like folding up on itself. So where are my keys? Okay, so here, this here represents right here, just this right here. Can you guys see can you see me? Okay, so this represents just the primary, okay? So then this here now represents our secondary, okay? So now we got alpha helixes, right? And then tertiary is just kind of folding it up so it all kind of fits together and does what it's supposed to do, okay? So tertiary is just the folding of the whole molecule and not just like regions, okay? So that's tertiary, okay? So primary, secondary, tertiary, okay? And then quaternary is like coming together of two different subunits. This is one subunit, my keys. The pen is another subunit, and it comes together to do some function, okay? So then that would be quaternary structures. Okay, now the beauty of quaternary structures, it allows this protein unit to serve multiple functions. So this guy could do one thing, and then later on it could interact with, let's say, my glow stick here and do something else, right? But still using one of those subunits. Does that make sense? So it allows for some flexibility in the body. Are you with me? Okay, how are we doing on time?
Pretty good.
Pretty good. Yeah.
Pretty good, yeah, pretty good.
Okay, so those are examples of our peptides. Okay, so let me give you a very specific example. Peptide that has a job. We call those guys enzymes. So enzymes, do you guys remember what enzymes are for? We talked about on Tuesday or Monday. No, maybe, yeah. Yes. So it basically helps us use less energy, okay? So enzymes helps us use less energy so it doesn't take us, we don't have to do as much work, right? So they kind of have a job and they basically do a little bit of the work for us. So in this context, we have two substrates. Here's substrate 1 binds to, these are called what we call our active sites, okay? Substrate 1 binds into our active site, this first active site. Substrate 2 binds into our second active site. Then it comes together. So this allows our substrates to come together. They found each other, and then they're able to make product, right? So once the product is made, the enzyme lets it go, and then we get our product, and it does whatever it needs to do. Okay, so these active sites serve a very key role, right? Because without the active sites, the substrates are not able to form, to bond, or to interact, or dock. Can't even think of the term. So often drugs that we use will actually block one of those active sites. So this guy, by blocking, is called competitive inhibition. Competitive inhibition. So it inhibits by competing. So in this case, since this guy now blocked that active site, substrate 1 can't bond to it. Only substrate 2 can bond, but it's not able to make the product. So often drugs that we use to sit there and, let's say, to lower our blood pressure, things like that, some of them are competitive inhibitors. And so they'll prevent the bonding of one of the substrates. Okay? We also have what we call non-competitive inhibitors, or allosteric inhibitors. Allosteric basically means that it binds at another site. So in this case, this guy will bind here, but it alters the shape of the enzyme so that this guy is kind of bent over a little bit more, and it keeps substrate 1 from binding. So that guy would be a non-competitive. So it's not competing for the active site, it just modifies the shape of the enzyme so that we can sit there and prevent substrate 1 from binding. So we have competitive and non-competitive inhibitors. Are we good? Does that make sense? And so a lot of the drugs that we use, like aspirin and high blood pressure medicine or protein pump inhibitors, all of those guys, basically play a role in either they're going to be allosteric inhibitors or they're going to be competitive inhibitors, competitive or non-competitive inhibitors. Okay, and they usually affect enzymes. Okay, so the last thing that we want to talk about is nucleic acids. Okay, so that's our fourth group. So I remember, if I remember correctly, we have carbohydrates, we talked about lipids, and we talked about proteins, right? Okay. The last one is nucleic acids. So nucleic acids, there are two types of nucleic acids. One of them is DNA, one of them is RNA. I'm going to tell you the distinction between the two to kind of help you. Well, one thing, DNA, deoxyribonucleic acid, has removed one of the oxygen. So it removed the oxygen from here. So deoxy, that's what D means. It means removal. So removal of oxygen. Okay? Ribose nucleic acid or RNA, right? So that guy has that oxygen there. So by the removal of that oxygen, it allows DNA to be much more stable than RNA. Okay? So DNA is more stable than RNA. DNA in our body holds our genes, the coding information. Right? It's kind of like the blueprint for everything. And you don't want your blueprints to get all screwed up. Where ribonucleic acids, they're sending out, they're like a photocopy of those blueprints so you can build what you need to build. Okay? And we'll talk more about that a little bit later. So let's see, what was it? Is this why some viruses can be killed easily? Yes, this is why some viruses can be killed easily. Some of the retroviruses are RNA viruses, right? The difference is that retroviruses, which are typically a little bit harder to kill than, let's say, other RNA viruses. So there's other RNA viruses that are really easy to kill because they don't make DNA. Retroviruses make DNA, and then that DNA integrates into your genome. So it's like a double-edged sword. Not only does it make DNA, that DNA will go into your personal genome. So you're kind of keeping a copy of it and keeping it alive. So for instance, HIV is a retrovirus. COVID is a retrovirus, right? And so those guys, they actually will integrate. And so it makes it a little bit harder to get rid of those particular viruses that others. So, but that's actually a good question. Okay. So your DNA is going to utilize, so the structure of DNA is going to be a phosphate group or phosphate groups. It's going to be your sugar, so one of those sugars, and it's going to be a nitrous base. Okay, so one of these nitrous base. Okay, so we have 5 different nitrous bases that are used for your nucleic acids, right? Cysteine, thymine, adenine, and guanine. These guys are all used for DNA. Is anybody colorblind? Jesse, are you colorblind? No? Good. Anybody else colorblind? So we assume that they're not. So I'm going to draw those in orange. Okay? So the ones that are for RNA, can you guys see the difference between the colors? So that's going to be adenine, guanine, cysteine, and uracil. Okay, so these are the ones that are going to be used for RNA. Okay, DNA will often be double-stranded. Oops. Meaning that you have one strand that is covalently bonded, and you have another strand that is covalently bonded, and it comes together by hydrogen bonds. We'll talk about that here in a second. And RNA is normally single-stranded. We already said that DNA is long-living. relatively speaking, of course, longer living or more stable. Let's just say more stable. And RNA is less stable. Okay. Questions, concerns, cash, credit. So nucleic acids or nucleotides are composed of the nitrous base, the sugar, and phosphate groups. Okay. Okay, so this is an example of DNA. So this is DNA forms what we call a double helix. So it's two strands that are covalently bonded together by the sugar backbone, sugar phosphate backbone. This is one strand here, and here's the other strand. Okay, and they actually form a helix, and the two strands interact with each other by hydrogen bonding. So if it's single-stranded, does that mean it doesn't twist like DNA? That is correct. It doesn't twist like DNA. So it can actually form loops and things like that by interacting with other structures on itself. So often a RNA molecule will interact with itself, like it'll form an RNA case of 10, where these guys are interacting. That lines represent the hydrogen bonds between two nucleotides. And so, or it'll form like a little helix structure, really long. And these guys will actually helix. So you'll have a loop and a helix going on. Okay. If it's long enough, but it'll only interact with itself and not another strand. Okay. Okay. Good question though. Okay, so the structure of ATP. You guys know what ATP is, right? Well, ATP is cellular energy. It's the energy that's found inside of the cell. And so all of the energy is basically stored up in the bonds. Primarily, it's stored up in the phosphate bonds, right? So you have ATP stands for adenosine triphosphate, meaning 3 phosphate groups. 1, 2, 3. These 2 phosphate groups store a lot of energy. The very first one stores the most energy, okay? The very last one has very little energy stored up in it, okay? So adenosine triphosphate is broken down to adenosine diphosphate, and then you get what we call an inorganic, we call it PI, or inorganic phosphate, okay? So when we break that bond, we release energy. That's energy that we can use for different things. So enzymes will often use energy to do their job, right? And they'll be breaking these bonds. So adenosine diphosphate can then break down and also release its inorganic phosphate. And then we use that energy for something else. And then the same thing within adenosine alone. Okay, so this is adenine. Adenosine is a combination of just the adenine and the sugar. Okay. Questions, concerns? Okay, I forgot one thing. Let me go back. Okay, so when we're talking about our double helix, adenine pairs up with thymine. So in terms of the two strands interacting by hydrogen bonds, so A matches up with the T. And so these guys are covalently bonded. A, C, G, A, A. So C pairs up with a G. So that would be a C, and that would be a T, and that would be a T. Okay, so they'll pair up together to sit there and to form the other side of the helix. So they find each other, and then they pair up with each other once they're made. Okay, and we'll kind of talk about how that happens later on. Any questions? Did I forget anything? Now, you can't be tired yet. We're going for the goal. Okay. Blank space. I have questions for you guys. You guys ready? Okay, Soledad, are you there? Okay. So my question for you is, how many different nucleotides are there for DNA? I'll give you multiple guess. So we have A would be 2, B would be 3, C would be 4, D would be 5, and E would be 6. What do you think, Soledad? 2? 2 is incorrect. The correct answer is 4. We have the A, the C, the G, and the T. Okay. So we should have a little competition. You think? A little competition? So Soledad.
Like fun.
Yeah. So we'll do Soledad. So right now, Soledad, you're down. Okay. So we have Alejandro.
Oh no.
Okay. Alejandro. So your question is,
Winner gets to do homework.
RNA differs from DNA by if you give me one of them, you can get 1 point. If you give me 2 of them, you can give me 2 points. See, extra and getting rid of, getting rid of oxygen. Okay, so, and what do you mean by an extra is it an extra nucleotide or is it an exchange for a nucleotide? And if you can give me that, you can get 3 points. That is incorrect. The correct answer is RNA uses A, C, G, and a U. So it replaces that T for a U, where DNA uses A, G, C, T. So you get 2 points. So you want to know what your guys are playing for? Anyone?
I want to know.
2 points extra credit going towards your test score.
Damn.
How about that? Is that pretty cool?
Can I start playing?
Okay.
Okay.
Okay. Adrian. For 3 points. Name 3 lipid structures. Are you there, Adrian? We'll count to 5. 1, 2, 3, 4, 5. Okay, the question goes to Soledad. Soledad, since Adrian isn't there, can you name 3 lipid structures? Are you there? Oh, there she goes. Triglyceride, that's 1. So we got 1. Saturated fats, that's kind of like a triglyceride. Unsaturated fats, both of those are those are both triglycerides, the types of triglycerides. You can do it. So those both would fall under triglycerides. They're distinguishing between those 2. Any guesses? Come on, you can do it. I know you can do it. Think about it. One of them starts with a P. Okay, that's 2. One more for 3. So we'll give you 5 seconds. 5, 4, 3, 2, 1. There we go. That's your 3rd one. Steroids. Okay, so Soledad has the upper hand. Okay, Alejandro. So actually, we'll do both of you guys. Alejandro and Soledad. So what I'd like you guys to do is, I would like you guys to think about the question first, type in your answer, and then when I say submit, submit your answer. Okay, so here's the question. The question is, why does water dissolve salt? Okay, so go ahead and type in your answers. So this is worth 3 points for the win. And if Adrian is there, you can also submit it for those 3 points, Adrian. Okay, are you guys ready to submit? 5, 4, 3, 2, 1, submit. So the answer technically is not quite that. There's an aspect of covalent bonds, true, but what aspect of covalent bonds that makes water able to dissolve salt? What type of bond is it? I know it's covalent, but you got to be a little more specific. It doesn't have to do with the fact that it's stronger than the ionic bond. You guys are, it's a little off. So think about it like this. So we have sodium plus Cl minus. What does water do? Hey, baby, what's your name? Oh, sodium. What does water do? Water is a home wrecker. That's right. It's a home wrecker, and it does what? Here's water. Here's water. Because water is polar. It's able to sit there and pull away the parts. Remember, partial positive, partial negative. So the partial positives all surround the water. All right, remember that? Let's see. Right here. You guys see it? Okay, so the winner is Soledad. Soledad, you'll get 2 points. Alejandro, because I'm a nice guy, I'll give you 1 point. So just can you do me a favor? Remind me to do that. Add a column to extra credit column too, bro. Then we'll have the same competition with the morning group.
Yeah. No. What is it? Unless it's back up, then this is irrelevant. What I'm gonna say, but
Canvas is down.
Yes.
Yeah, I know. That's why I said remind me. Yeah. Okay, so I have nothing else for you. You guys are free to go. You guys have simulations to do. If you have any problems, you can contact me or Jesse. My office hours are Thursdays at 1 to, no, not 1. Thursdays at 2:30 to 4:30 and 7 to 9 on Thursdays. Or you can text me or remind me. I will eventually get back to you. I'm a little slow some days, but I'll try my best. Okay, so then I'll see you guys next Monday. Have a good weekend.